Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I explore advances, challenges, controversies, and opportunities in brain imaging and modeling with neuroscientists from around the world. Today, I had the pleasure to talk with Dr. Max Shine, a tenure-track University of Sydney Robinson Fellow and a systems neurobiologist working to understand the mechanisms of cognition and attention. He received his bachelor's in biochemistry and psychology, his medical degree, and his PhD from the University of Sydney, and also was a postdoc with Russ Poldrack in Stanford from 2014 to 2016. Mac has been creatively forging new approaches and correspondingly new insights with functional brain imaging and modeling, specifically how the brain dynamically updates and reconfigures during resting state and activation. He's deeply interested in subcortical control of cortical configurations as well as arousal. I've been so impressed with how he is employing entirely novel ways of analyzing fMRI data, and through his modeling and multimodal research, providing insights into subcortical-cortical coupling as the nexus of brain function in healthy and clinical populations. Our discussion jumps from nuts and bolts to open questions and challenges, to high-level questions and perspectives on the limits of fMRI and modeling. This discussion is packed with insight and was truly a pleasure, as Mac is both so eloquent and passionate about his work. I hope you enjoy it. Okay, welcome to the OHBM Neurosalience podcast. I'm Peter Bannattini, and today I have the honor of being speaking with Max Shine from Sydney, Australia, University of Sydney. And I recently, it feels like recently, it was actually almost 6 months ago, came back from OHBM, where he gave a keynote lecture that was just brilliant, sort of laying out his view of using fMRI and other approaches to I feel like, in my perspective, at least, of your talk was, it was sort of reintroducing, obviously there's many types of analysis of fMRI data, but reintroducing, sort of looking at it through brain states and also sort of tying in subcortical systems into the story, and which really, I think should be done more as far as that's both subcortical systems and novel processing techniques for looking at, like, for instance, integration and segregation. And we could talk about that as well. So, and also, of course, I knew, from when you were a postdoc with Russ Poldrack, and just an amazing couple of amazing papers you wrote with Russ, and then now you're continuing on your tear of doing great work. And specifically, actually, and like I said, we can talk about this more. I feel that the field's been shying away from even thinking carefully about subcortical systems because, at least with fMRI, it's hard to image, and it's hard to image in many different ways. I mean, EEG, MEG, you don't really know what exactly you're looking at, and so and everyone's been sort of posing their questions around their tools. And I think we're missing, in my opinion, and possibly right, the most important part in some sense. So to begin, all right, sorry, welcome.
Thanks, Peter. Thanks for having me. Real pleasure to be here.
Yeah. So just to begin, I'd like to always start the podcast with the person's history about kind of what motivated them, what got them started. So. What's you're both a clinician and a PhD.
Yep.
So what got you interested in the brain in the first place, and and specifically neuroimaging, as far as that's concerned?
Yeah, it's a great question. So I think I can kind of track an interest in the brain, back through early high school years, not in any really sophisticated way. But so much so that when I was in undergraduate in Sydney, I took a bunch of psychology and neuroscience courses and found it just fascinating that you could kind of link something as nebulous as our behavior, our experience of the world, to this kind of organ system in the brain. And so I later went to medical school, learned a ton about systems biology, thinking about how all the different elements of the body work together, how the fact that the heart pumping blood around the body is deeply connected to whether or not the lungs can actually extract enough oxygen from the air. And then your kidneys have to be able to filter out all of the different byproducts of your of metabolism in order for you to function properly, for your heart to pump it the right way. So thinking of this really kind of emergent systems level perspective was really deeply informative. But the moment you got above the neck, all of a sudden, all the stories just became purely descriptive. It would be something like, oh, I know that chronic heart failure happens because the pressure in the different chambers of the heart becomes abnormal, and now you get this backflow. But what about Parkinson's? Oh, that's just the basal ganglia. Turn it off, right? And you would just have these sort of stopping points where you were kind of expected to take this really rich process-based explanation and just put it to the side and say, oh, that's too hard. And Parkinson's is really one of the bastions of explanatory neuroscience. It's really put up on a pedestal as a really great explanation where you can take a pathology in the brainstem, link it to circuit-based emergent properties in the basal ganglia and the cerebral cortex and thalamus, and then link that to symptoms. And so here was this place where you had this chance to do the process work, but to me it just hadn't been done. And so I developed a real, I want to call it a frustration, and I think we don't talk about this enough in science. I think a lot of progress comes not from inspiration, but frustration, where you say, you know what, I don't like that type of an answer. That's not satisfying to me. And as a resident working in the hospital, I kept trying to push for these process-based explanations, not just for something like tremor and Parkinson's, but the more complicated symptoms of neuropsychiatric disorders. Why is it that this person, can't count backwards from 100 by sevens, and that causes their feet to stick? You need to have these really process-based explanations of these kinds of weird and wonderful symptoms. And so I ended up transitioning into a PhD. I really vacillated for a number of months as to whether or not to do this, because it's a bit of a risky step going away from this really kind of obvious career trajectory in medicine towards the kind of nebulous world of academia. But the moment that I'd stepped into that world, it was like, yes, this is for me. I just love the fact that in academia you get to not pretend that your uncertainty doesn't exist, but just live in it. Like, I don't know what this is, and I'm going to try and figure it out. And that I think in my core, I'm a real puzzle solver, and that's something that's really deeply resonates with me. So once I took a step and I didn't look back and did a PhD in Parkinson's and neuroimaging, and then off I went.
Okay, that's actually and right. I mean, it's interesting with the brain. I mean, it's obviously, I feel like, right, compared to other areas of medicine, I it's always embarrassing to say with, I work in NIMH, National Institute of Mental Health, and I say, we're, mental health is kind of in the dark ages in some sense. It's, the equivalent of like, bleeding people with leeches when they're sick, because, we really, the brain is this system that we obviously are trying to probe into the circuitry, and we tried drugs that we kind of know what they do, and they seem to work, and it's very empirical, and so it's there's a long way to go, and I think we have to do whatever we can to start digging in and addressing the problem, like you said, getting frustrated, and
just to add on to that, Peter, I think that's deeply right, and I think that one of the benefits I think that I've sort of tried to keep with me coming from medicine into a really kind of rich blue sky world with just so many brilliant people. I quite love working in modern neuroscience with the neuroimaging and systems neuroscience, because it's so diverse in terms of the backgrounds of folks that you often have to kind of stop and appreciate just how amazing it is that we can even talk to one another. I mean, I have conversations with physicists and engineers and mathematicians, and I've got no training at all in those backgrounds. But the reason I think having that clinical background is so important is that you kind of have been exposed to all of the different ways in which the thing actually works and breaks. And you can't kind of have these halfway house stories, schizophrenia is just due to gene X. Well, you're like, well, I don't know, man. I've seen a lot of people with schizophrenia when I was a clinician, and none of them look the same. They look really different, and they've all got these really complicated stories and histories and recursion to old symptoms. Some of them can live with it for quite some time, and then it crashes. Other ones, it immediately destabilizes them.
Yes.
So you just realize immediately that a simple one-hit story is just not going to cut it. It's not written in the same language as the disorder's written. And so I think clinicians have got this capacity to tap into that. And so I think having conversations with clinicians is deeply important, especially if you're a mathematician or an engineer or someone that is really good at speaking linear algebra, right? We all have to do this in neuroimaging. Clinicians don't get any training at all in linear algebra. So you've got to find a way to connect with them so you can learn what it is they're trying to explain and try to understand in the language that you know is one of the most beautiful forms of mathematics we have. And so I think there's a real challenge for us that as a field to kind of create those bridges. And I'm looking forward to being part of it going forward.
Yeah, I think that you're right. I think actually communicating and having people that are good at encapsulating, concepts so that people can actually, get an intuitive sense of what the nature of the problem is. And, along those lines, I mean, sort of jumping ahead a little bit, and this might be relevant to some of our discussion. I heard a really nice talk by Nicole Rust. She recently talked about the problem with understanding psychiatric problems or even looking at the brain as a. She was saying, in her analogy, and I don't know, and this is interesting to see how far it applies. I mean, so she's like, well, certainly the brain is this complex dynamical system, and we can predict it on a statistical level, but you know, just like the weather, for instance, you can't predict more than a few days ahead, and you certainly, and even if you had means of like tweaking it here and there or trying to adjust some aspect of it, because it's a complex dynamical system, it becomes, it's becomes hard to predict beyond a certain amount of time, and that's the question is, let's say we completely understand how the brain works in terms of, and we can model it very well. Is there still hope for therapy in some sense? Let's say we can even tweak the circuitry. Like you said, everyone's somewhat different. Complex systems sort of manifest themselves in different ways. Is it predictable? Is it predictable enough to be able to? It's not a linear system in that regard.
Yeah. So, I mean, I've got a few things to say about this. I read Nicole's paper in the Transmitter. I loved it. And I think that using weather as an analogy has a lot of really deep lessons to teach us. One is that causality in complex systems looks kind of weird. And it's a little different than the kind of traditional billiard ball type of causality that we get taught about. A leads to B, leads to C, leads to D. And if that happens, then the reflex arc has been completed and you complete the action. In complex systems, particularly multi-scale complex systems, there's these really strange things that happen where the emergent property of a system, let's say the kind of lower level features all combined together into some higher level feature, now has an impact on the types and the probability distribution of the lower level systems activity. So a really nice analogy that George Ellis used was two people with identical genetics could be born, let's say they're identical twins, they're born, and then one of them is moved into Calcutta and the other one's taken and put in Rio de Janeiro.
Yeah.
Those two individuals, completely identical genetics, will end up being completely different people. And even if they have similar personality traits, they'll speak a different language exactly because, and for no other reason, that they were in a different context. And so it's very, very difficult to think about this kind of a system in the same billiard balls way, because all of the same billiard balls are being followed, but they lead to completely different types of outcomes. So we need different types of stories and methods and frameworks to try to understand what causality looks like in that kind of a system. One that we've been leaning on more and more is kind of the dynamics of control systems, where the emergent sort of output of a system is then fed back into the network as another ongoing data point, which then the system has to deal with. And I think this is a kind of truism of brains. Every time we take a step forward in time, we're now dealing with both the input that's come in plus the machinations of the internal system, maybe some anticipatory dynamics, maybe the system we needed to do the processings in a refractory period, what have you.
Yeah.
These details all deeply matter for the function of the system.
Yeah.
And these types of billiard ball causation methods we have don't really take this into account. So, we're not alone in trying to tackle this. It's a really hard problem, but there's lots of ways forward for it.
Okay.
The second thing that I think the analogy can teach us is something my wife taught me. My wife's a medical doctor, and we chat about work sometimes. She often yawns when I start talking about the thalamus or the cerebellum or something. But one time I was talking to her about this very analogy, the idea that you could take a weather system and you could have measurements of the temperature in different suburbs, and then you would say, oh, because of the humidity and the temperature and the basic wind flow that we can predict, it ought to rain at this probability. Or our simulation suggests that in 50% of simulations it'll rain tomorrow. And she said, and I was lamenting, the brain's so complicated, it's just like weather systems. We'll never be able to predict the next step. It's too complex. And she said, well, actually, it turns out there was actually a study that was done that they were taught about. She's actually from the States, and there was a study that was done back in the day where at Cape Canaveral they put a weather balloon high up in the atmosphere where it could detect really, really rich data over a large area. And the weather balloon actually got such pure data that its prediction horizon went way out, and it was able to actually accurately predict the weather for something like a week ahead of time. And it was really great. Now, I don't know if this would still work in our climate change time where variability abounds. But it was a really important lesson, I think, which is that sometimes what we are actually saying when we say that the brain's not predictable is that our measurements actually aren't sophisticated enough or trustworthy enough to actually make those strong predictions. Because what happens is that in these prediction models, because there's so many of those different feedback loops that are playing in on one another, small differences start to constructively interfere with one another, such that your prediction horizon becomes so wide that you don't know exactly what's happened. Yeah. And so I think that there's something, there's a really important lesson for us all there to essentially keep pushing the methodological front. Let's try to keep getting better and better data. Let's try to keep seeing if we can get the TSNR down. Let's get different methodologies. Because and we shouldn't essentially throw our hands up and say, oh, because the methodology couldn't do it, the brain couldn't do it, and confuse the map for the territory. I think there's room to grow there as well. Right.
And I do think that, there's different types of prediction. Obviously, it's hard to predict a person's, on certain timescales, but it might be easier to predict general outcomes of, improvements or not. Who knows? Okay.
I can actually say one more thing on this front that is kind of fun. So a really talented postdoc in my group, Eli Muller, who has a background in physics, has been with me for a number of years. He's now a research fellow at the University of Sydney, working on some really, really interesting problems in this kind of interface between physics and nonlinear control and neuroimaging and neuroscience. So we've sort of struck up a collaboration with Steve Brunton and Bing Brunton at the University of Washington, and they're kind of dynamical systems experts. They think a lot about the mathematics of dynamical systems and nonlinear control systems. And that's Steve. And in Bing's case, she does these really beautiful experiments on biological systems. Like, how is it that a little fly can detect a change in wind gradient if it's trying to trace a scent down to find, let's say, a piece of food, and all of a sudden the wind shifts. What's the optimal trajectory for that fly in order to make sure that it stays with the wind? So really, really fun people to talk to. And it turns out that this problem of trying to predict into the future is actually a really well-traipsed problem in a lot of nonlinear control systems. And one in which Steve has done a lot of work in is fluid dynamics. And so you can imagine, I want to, as a plumber, figure out whether or not I'll get lots of turbulence in the pipe system that I've set up, or whether or not it'll flow really neatly. So that, and this might be really important for the efficiency of an engineering system, right? Like a factory or something. And so what you can do is you can apply you can measure the flow in the pipes, and you could figure out how laminar they are, how much they flow versus how turbulent they are, based on the kind of, what you do is essentially look at the update, and then you do an eigen decomposition of the update matrix, and you look at the eigenvalues of that, and you get lots of information from this.
Yeah.
So Eli thought, well, what if I do that to human imaging data? Just, really nice 7T resting state data set we got from Luca Cocci and colleagues. And let's just fit a really simple update matrix and see whether or not it is stable and laminar or it's turbulent. And the really fascinating answer that he found was that it's actually kind of both, in that there are moments in the recording when the flow is really laminar. It's really predictable. You can tell exactly what's going to happen for 10, 15 seconds. And then there'll be this abrupt change, and then it'll shift to something more turbulent. So go on.
But let me just ask you, back up for a second. That's really interesting. So how do you actually, so you use laminar versus turbulent in terms of fMRI data. Could you break that down just a little bit?
Sure. Yeah. So you take the data, you essentially calculate like an autoregressive model in the data, just an AR1. So you're asking for every single region. In this case, we use, I think, the Schaefer parcellation, the 400 parcellation. You're actually asking at each point in time, what's my expectation of what the bold signal will be in the next TR for that particular region, and I do that across all the regions. And then what I do is, at each point in time, I then say, how good was my estimate? So if I go forward one point in time, I know the current value, X, of all the values, and I ask what's it, X, T plus 1, and then I sum up the difference between my guess. And then but then importantly, I don't just do that again. What I do is I take my estimate and I go forward another step. And then I take my estimate and go forward another step. And I essentially go out on my little forecast horizon. It's a little bit like saying, I predict today that the weather will be sunny for 3 days, and then it will rain for 4 days, and it'll be sunny again. And I go through time and see if I was right. And it turns out it actually rained on the 3rd day, but it was sunny for days 6 and 7. So I get all these errors, and they start to stack up over time as I get wrong. What you do then is you slide that all the way across time. So then for every TR plus the horizon of 10, I now know how well I was predicting. So you can then imagine taking a slice of that hilly landscape. And if the data was completely predictable or laminar at all times, what that would mean would be that at every single point in time, I should just immediately be able to tell you what the next time point would be without any problem. And given the sluggish nature of bolds, the TR here was, I think, 0.58. So we'd expect there'd be a lot of autocorrelation. You're pretty good at TR equals 1. You're pretty good at TR equals 2. But what you start to notice as you extend out into that forecast horizon is that there's valleys in this landscape such that even out to 10 TRs into the future, so 5 or 6 seconds, there's still really, really, really lovely predictability for quite a period of time. But then all of a sudden there's an abrupt change. And now the bold signal at time point X was absolutely not predictive at all of time X plus 1 or X plus 2. And you see these really, really, really interesting fluctuations between laminarity and dyslaminarity. To cut a very long story short, and maybe it'll be helpful for coming back to this later, we can actually link those periods of stability, laminarity, or instability and dyslaminarity to a particular organizing feature of the thalamus, which is that this deep structure, which is connected to the whole cerebral cortex, has these different types of projection patterns. It's really a spectrum between these two extremes of really targeted core-like projections and diffuse matrix-like projections. And it turns out that the moments of stability are really related to the activity in the core nuclei, and the moments of instability to the matrix nuclei. And we use a bunch of approaches and some modeling, and then an actual direct stimulation of a macaque's thalamus in the matrix thalamus to test this hypothesis. So there's some really, really lovely, rich multimodal evidence for this. And I think the reason I bring that up is to say that at the end of the day, I think the crucial thing for us to do as a field is to take these really lovely abstract concepts from nonlinear control in physics, but actually ground them into the actual physical neurobiology of the brain. Like, here's what the brain is. It's this weird system full of neurons all connected together. Why is it organized like this and not some other way? And I'm really interested in understanding that kind of intersection between those two extremes.
Yeah. And so actually, that's actually right. I love your approach because, I mean, part of what fMRI has, sort of evolved in the sense that, initially we were just simply looking at, do a task, see where activation is, and sort of mapping out where, it corresponds to behavior. But now, and then, we go along and we, um, parselate the brain, look at resting state, and see what's, functional correlation, for instance, or resting state correlation, and or even, but then trying to then keep going, and like you said, look at, bring in the concepts of laminarity or turbulence, and and sort of try to tie those into, rethinking about how, right, how the thalamus controls things. And yeah, I was going to ask about that. So, why the thalamus, not the locus coeruleus, or other subcortical systems. And you just mentioned that you tie this in with multimodal sort of animal work, which is beautiful. And I
So
really like, yeah, go on. Yeah.
we did that too. It just didn't go in the preprint. We have another collaboration with Valerio Zerbi in Switzerland. He's now in Geneva, who had this brilliant study where actually back in, I think it was 2017, they had a paper in Neuron where they actually had created a genetic line of rodents that actually had a DREADS receptor. This is a designer receptor which you can now turn on and off with a ligand that isn't used as a ligand anywhere in the body. And so you can essentially, like, on purpose, silence or excite a particular
Oh,
area
wow.
of the brain with control. But importantly for us, he actually measured whole brain bold in these rodents. They had to be lightly anesthetized, so you always have to take this work with a slight grain of salt because anesthesia does have an impact on the kind of resting dynamics of the brain.
Yeah.
But the really cool thing you could do is you could go in and you could actually mess with the locus coeruleus and turn it on and off. And he essentially found some really lovely evidence that was consistent with some of the stuff that I'd done with Ross Poldrack in my postdoc, which was that
Yeah.
the ascending arousal system can kind of control the communication or the sort of interactions between different parts of the cerebral cortex, and specifically for our work, in a way that renders them more sort of integrated, is the term we use in the framing of network neuroscience. But the intuition is that it breaks down kind of the standard, I don't want to use the word communication, the standard covariance patterns that you see in typical resting state data, and makes these really interesting novel combinations. And
Yeah.
so what Valeria has done in following work, because the problem with DREADS is great, but the problem is it's a light switch, right? You're turning this thing on or you're turning it off. What you really like to do is come in and be able to turn something on and off really quickly, or turn it on really, really at a high frequency or a low frequency, and really see if there's these sort of dose-dependent effects.
Yeah.
And so what Valerio did in a follow-up experiment was actually use optogenetics, which is kind of like DREADS but on steroids. Now you're using a light which you can turn on and off to excite this nucleus, again in lightly anesthetized animals, again
Yeah.
with whole brain bold.
Yeah.
And what Valerio and Christina, the first author, did in this really beautiful experiment was actually show that you can get a kind of dose-dependent effect of the locus coeruleus turning on and getting this really interesting whole brain connectivity pattern that arises. So you now have this really rich causal evidence for
Yeah.
something that we had to kind of infer indirectly as a hypothesis back when we were working with humans.
Yes.
But anyway, we've run the same thing in that data as well, but we haven't put in the manuscript. I think sometimes this is a slight problem with systems neuroscience, is that because the brain, everything's connected to everything, you end up using the same vantage point or the same analogies to explain the impact of particular structures. And I think
Yep.
this is actually a kind of an important point for us to grapple with. Brains are incredibly redundant in a lot of ways, and that what you really don't want to do is have the loss of one particular neuron just devastate the system.
Yep.
And this is really what happens when in forms of dementia, right? If you lose parts of the arousal system in Parkinson's, if you lose the dopaminergic neurons in the substantia nigra, if you lose the basal forebrain in Alzheimer's disease, you start to see this massive, massive loss of functionality from the loss of a very small number of cells. And
Yeah.
so these kind of choke points are really, really risky for a big complex system like the brain. And I think in areas like the cerebral cortex and the thalamus, this kind of has been avoided a little bit over recent phylogenetic time.
Yeah.
So I think this redundancy is actually kind of a feature rather than a bug.
Okay. Interesting. So that's actually, I mean, it brings out a good point of how, how fMRI sort of like, has a place in sort of motivating sort of hypotheses that can be tested further in some way. And so, you see
Absolutely.
this sort of effect, and you said like you form an hypothesis, and you go in and test it. And it's also interesting too. I mean, so along the lines of looking at liminarity versus not, but also, you mentioned briefly looking at trying to assess integration versus segregation in the brain.
Mm-hmm.
Uh, and I found that, I was reading that paper, and you said, oh well, and it also seems that, it seems to correlate with intelligence, like, putting, breaking things down into their lower dimensional, just taking ICA and then seeing the manifolds that appear, and it seems like you were saying that uh, uh, it seems to correlate it with intelligence. I'm probably hacking this, but that the uh, the uh, integration is I mean, the integration state is sort of more cleanly manifest in some sense, and I thought about that. I'm like thinking, well, there's other work that actually parcellates the brain and looks at areas that show more tight correlation, and
Mm.
there's similarities, and that's the beauty, and also the problem with fMRI is that there's different ways of approaching this sort of question, and one sort of puts it in a framework of integration and segregation. Another one puts it in a framework of, oh, these networks are more tightly correlated with each other. And then I thought,
Yep.
well, the only way to unite those would be, maybe the network that's tightly correlated is broad, and so it looks like broad integration,
Yep.
and it's a big
Yep.
network and dominates the first ICA component or something.
Yep. Yep. I think that's right. We haven't done the kinds of experiments I think you'd need to do in order to determine whether or not you could actually make these strong conclusions about these different frameworks. What I will say is that the point you mentioned about different methodologies and different approaches is deeply important for us to grapple with as a field. And we in my group try really hard to take what we call a pluralistic approach, which is to say no method is worth anything more than any other. They all have their own set of assumptions. There's no free lunch. You can't get a pure look at a nervous system in action.
Yeah.
The best thing you can do is to have predictions that are independent from the recording modality and independent from the algorithms that you're using to test it, and then try to find the best algorithm or recording modality that matches the resolution of your question. So this again is one of the reasons that my group has really shifted into using computational modeling a lot. I was really inspired by working with Michael Breakspear in the kind of latter part of my postdoc, and then subsequently developed a really strong collaboration with him along these lines. He really convinced me that if you look back at the history of physics, a lot of the huge uh, inroads that have been made into understanding the kind of fabric of the universe have come from an appreciation of our problem, where we were thinking of a problem as something just because our observation model was not sophisticated enough yet. We
Yeah.
used to think that the Earth was the center of the universe because that was just an assumption that we made
Yeah.
until we changed our observation model and thought, what if it's something different? What if it's not, Earth-centric, but heliocentric? And all of a sudden now the mathematics could make sense in a way that they didn't beforehand. And I think
Yeah.
in our field, at least when I grew up in the human brain mapping community, I think the challenges associated with analyzing fMRI were more than enough to keep me busy for a very long time. And I'm still learning things about bold data and the types of stats we have to use and all these things, that it keeps me very busy. So I think it's a hard challenge for a young student to essentially say, what do I do? Do I dive into the complexity of this field and learn all of its nuances, all of its hard-won lessons? Or do I try to distribute out and do a cool experiment like Todd Constable had a really beautiful paper where they did simultaneous calcium imaging and bold imaging
Yes.
in rodents. I mean, that's an incredibly difficult paper. Evelyn was just a hero for taking that on, but the insights we get from it are brilliant and really push the field forward. But you talked to Evelyn about this, and she's like, yeah, it took a lot of time to get good enough recordings of both the bold and the calcium alone, let alone them together. That
Yeah.
was, years long effort. And it was a real a choice for her to do that rather than some off the shelf fMRI data set. So I
Yeah.
think these are really, really difficult problems that I don't really know how to tackle as a field. Well, we've got to find ways to support people for taking these choices into these kind of multi-domain spaces. But we also need to kind of appreciate it's going to take a lot longer to do it at a high level that we can trust. And anyway, I just wanted to I think that's an important point for us to grapple with.
Yeah, no, it's actually, I mean, they're right. I mean, I think it's a mistake to be sort of tied to a method and sort of like, just keep on pounding on that method, because I think it's understandable to start that way. I mean, you're trying to
Oh,
learn
of course.
something and you have to do something, uh, but yeah, I mean, trying to then, get a deeper understanding of what the methods are trying to probe. And so to start to generate the questions and then see how the methods could best address it. Like even when you were talking about laminarity again, I have to go back to that. And I was thinking,
Mm.
oh, well, maybe, there's a former student of Olaf Sporn's who's now my postdoc, Josh Vaskowitz,
Oh,
who
yeah.
does edge analysis. So he
Yep.
does, he looks at the edge time series. So, each moment by moment, it's sort of like CAPS analysis where you look at the moment by moment changes, and maybe that could pull something out. That's, you're trying
Hmm.
to basically tie the methods to the nature of the data, but also you want to sort of tie the methods to something that can give you a deeper understanding of maybe the principles or
Yep.
the dynamics in some
Well,
sense. And yeah,
to,
it's a
to,
good.
to bring that back to the modeling again, just to kind of close that thread. This is where modeling is so useful, right? If you take that old Feynmanism, if you can't build something, you don't understand it. Well, here's the opportunity, right? You say, I think that these kinds of dynamics that I'm seeing are because the thalamus is like this and it's connected to the cortex like that. Um, Build a model with different constraints,
Yeah,
have it give you an estimated bold time series, and then see whether or not your prediction from analyzing that data is the same as what you see in the empirical data. So, and ultimately, it'd be great if you could take the edge time series, if you could take integration segregation, if you could take laminarity dislaminarity, if you could take some of the energy landscape stuff we're doing, hidden Markov models, whatever you want, and you apply that all to the simulated data, and then hopefully what you'll see is actually that it doesn't match some of them, but it does in others. So now
yeah,
what you've got is a chance to update your model. What are you
yeah,
missing? What part of the nervous system have you not put in there with your assumptions? Because all of these models are really massive oversimplifications of
yes,
the complexity of the brain. So
yeah,
ultimately, what we're really looking for, I think, as a field, and we try to really bring this to the core in my group, is to essentially say we're not looking for answers that stop the conversation. What we want is an answer that leads to the next best question that we could ask. Sometimes that's we need better data. Sometimes that's we need causal control. Sometimes
yeah,
that's we need to go read old anatomy textbooks and figure out what on earth the posterior versus anterior intralaminar nuclei of the thalamus are doing. Like, why are they like that? And I love those parts. That's my happy place, is reading old neurobiology stories and trying to understand the complexity. But the physicists in my group, they love simplicity and they love elegant models that capture all of the different elements of that you're seeing and show you that it's actually a property of a really simple process. And so I think having that tension for us is so crucial. But at the end of the day, it's all about trying to find answers that open up new vistas rather than trying to find answers that is
yeah,
we've got unimpeachable evidence that this is the right way. It's like, no, man,
yeah.
science is a
Change everything.
process. Yeah, it's not a set of facts. Anyway.
Yeah. No, that I totally agree. And actually, I was just about to bring up that I often when I look at I see Michael Breakspeare's beautiful movies and I think to myself, okay, so like you said, uh, uh, is it such that I'm even amazed that the models work at all in terms of predicting, because fMRI, I mean, admittedly, is you're looking at millions and millions of neurons within each voxel,
Yeah,
and
it's
there's
astounding.
obviously organizational structure all the way down,
Hmm.
and it seems that those scales, we're looking at those, some sort of, uh, you know, combined action from all those scales, all to some level where we can predict things,
Yep.
and and uh, and it works. I mean, we can actually then simplify the model such that we can predict that. To what level does that give us insight as to, you know, what's actually going on, the
Mm,
mechanistic,
mm,
you know,
mm.
excitatory versus inhibitory activity, what are all the neurons doing, and how and why are they somehow behaving in a way that we can explain at this sort of temporal and spatial scale? Uh,
Yep.
are- is there hope in sort of understanding this deeper scale? With obviously we need to the measurements, but is this modeling useful? in some sense, in terms of
Yeah, no,
yeah,
I think your point is spot on. It's we haven't got the right kinds of analogies, I think, to even understand how to think about a system as beautiful as the brain. I think about this sometimes when I'm a big sports fan, and I think about, trying to imagine what was inside someone's brain before they made a big choice to, cut to the right versus cut to the left. And if you think about trying to explain it like that, you're like, man, there was just billions of neurons all interacting in just the right way, such that they coordinated the system to do this very specific movement at this specific time while the defender did this other thing. And you just think, oh my Lord, like, how are we ever going to have a reduced version of that that's satisfying? It's going to be, oh, that's just what happened in that moment. Like that's like saying a raindrop fell on this particular tiny, point in my backyard rather than it raining at all. We're not gonna be able to understand it in that rich way. But
right.
I think, part of my goal on that sort of side of my lab of understanding the richness of the brain, I think is destined to never succeed. And I'm so thankful that that's the
Yeah.
case. So what a beautiful problem to have for the rest of my corporeal life. On the other side of things, when I put my clinician hat on, I say the good news is we don't need to know the brain that well in order to be helpful, right? We don't understand how the heart and the liver and the kidney work at that kind of incredibly rich detail that we could put a specific drug in this location at this time to change the system.
Right.
No, we know that if we put in a calcium channel blocker, that generally means that when a heart starts to pump really, really hard, it can't do that as well. So now can't get pushed out of its regime, and now you don't get to run the likelihood of having a loss of blood flow and then a heart attack. So we
Yes.
don't need to have this extremely precise, rich explanation. We need better stories about how to intervene. And I think those are coming. If you look at the kind of forefront of the systems neuroscience literature, there are deeply intuitive stories about cross system interaction, cortico subcortical brainstem circuits that are really crucial for behavior X and not for Y. If we knock out this part computationally, it's doing this thing. If we put this drug in, it pushes them to this direction. So
Yeah.
these things are starting to happen in animal models. Like if you look at Drosophila, it's unbelievable the kinds of explanations that they're getting now. So I think if we can fast forward in time and we imagine getting better recordings of the human brain, but also trying to figure out the really difficult problem of how to interact with it in
Yeah.
a spatio-temporally precise way, then there are going to be wins, and
Yeah.
they don't require us to understand the whole thing.
Right.
But the understanding will deeply help because it'll show us where to look. So I think these things are deeply intertwined with one another, and there's hope. It's just a hard problem.
Yeah. Yeah. No, and I agree. I agree. I think there's hope, and yeah, I didn't mean to paint too grim of a picture, but I actually, because I'm actually always trying to get my head around modeling. For instance, you talk to Friston, and he has. It's not really a model; it's more of a construct of like
Mm-hmm.
the brain is doing predictive modeling, and that's pretty much can be summed up as that. You have you minimize surprise, and that's a principle, and it makes you feel like you understand something deeply, but you want to have models that then can predict you know how it's going to behave in specific contexts and what the mechanisms
Yeah,
are, things like that. But
yeah.
um,
Yeah, I think modeling can take all forms. Sometimes a statistical model is enough for you to do your work. A lot of times clinicians use statistical models. Not every person that gets aspirin is actually going to prevent a heart attack, but the odds are you will prevent one, out of 7 or something like that, heart attack events. I can't remember what the stats are now, but so these things are all very probabilistic. Carl's a genius, and I think one of the problems with genius is it's oftentimes very hard to understand how to apply the lessons that have been learned to the kind of coalface stuff that we're doing.
yes,
I will say I was kind of blown away, and I was actually chatting about this with Breaky the other night over a beer in Newcastle. If you look back at when just around the time that statistical parametric mapping really kind of took off, Carl was a huge had a huge influence on the field in terms of providing practical but really robust tools for doing this incredibly complicated multi-mass univariate stats on these data sets in a really nice way, and I think made it really approachable. I was the first software that I used when I started out as a grad student with no one else in the lab doing neuroimaging. And it was intuitive and you can understand it. Around the same time, I can't remember if it was a year later or a year before, he had a paper in Neuroimage where he was speaking about the idea of taking a completely different approach, which was doing dimensionality reduction on data using PCA and then tracking the trajectories of the PCs over time during tasks.
Okay.
And you're like, yeah, right. That's where we kind of, as a field, have now come around to and thought, yeah, this is actually a really interesting, intuitive approach for asking different kinds of questions.
Yes.
Now we're less worried about exactly where was the most extreme statistical abnormality in terms of the bold signal. Now what we want to know is, does the masked potential of the whole system have some interesting differentiation in it? And so I think it's funny thinking back historically to kind of some of the benefits we got from going down the SPM route, but also some of the corners that it painted us into. And I think now that we have this kind of plethora of different modalities, we can now start to say, let's put the brain in the middle, and then let's imagine looking at it through all these different lenses. And through an SPM lens, it'll look like this, and we'll get this kind of an answer. And what can we do with that? We can compare it to, meta-analyses. We can see whether or not people have lesions in those areas that also have problems in that behavioral capacity or something. But if we want to know how do we steer the brain away from this type of a pattern, this isn't going to help us, because this pattern we're seeing is due to both the pathology right now, but also the years of compensation that have happened since the pathology started, or maybe the kinds of things that you need to do to actually overcome the challenge. And so we've got this big, messy statistical parametric map. But maybe if we go back and do modeling, now we can say, oh, it turned out that having this lesion here of some subcortical system meant that now the rest of the brain had to compensate, and that painted you over here, and that's why you get the map like that. Yes. And I think the thing that is really important to me is that these two answers look completely different from one another, and they're both useful for different things. And so I probably come across as a little bit too optimistic, a little bit too inclusive, but I think this is important for us to grapple with. There's no one method that gives you the answers. It's about having the appreciation of the different vantage points and the strengths and weaknesses that come from them, and then figuring out how to have that conversation about which is the technique we need to answer our specific question. Yeah. And if you have that conversation before you start your experiment and you really get into it, I think you come up with much more satisfying answers and really enriching next questions.
Yeah. And I completely agree with you in every single way. And it's sort of, there's certain things that makes me think of, like, for instance, the Merrick paper that talked about, well, you need 2,000 subjects to draw any inference. And I thought that was a wonderful paper because of the fact that it kind of made us realize, like, okay, what's going on here? It's not, and I usually argue that it's not a statement about the sensitivity of fMRI. It's a statement more of how variable every person is. And
the kinds of questions we're asking.
Yeah, the kind of questions we're asking. If we're just simply putting all pathology into a group and looking for, some sort of biomarker based on resting, is that really the right way to go about doing it? And so maybe it's telling us probably not. We might want to look at each individual subject and look at their dynamic properties in some sense, and then collapse the data along those lines or something like that, as opposed to
sorry, I didn't mean to interrupt, but I'm too excited. I'm too excited about the thing I want to say, which is that if you're a student out there, this is where your frustration helps us, right? Because if you say, I agree with what Peter just said, which I think is a great summary, what do we put in its place? What does it look like? What is the process? What is the signature of that process? How do you help us find it in a way where we can trust that we've seen it and not some obvious noise-driven fluctuation that would look the same way? And this is the kind of inherent challenge that I think actually makes us as a field stronger. When I was writing a recent review with Emily, and a previous trainer of yours who I really love working with, and Russ, we really kind of wanted to bring out the fact that fMRI is a field that's kind of battle tested in a lot of ways, right? Like, we've made public mistakes before. Coke versus Pepsi, dead salmon, voodoo correlations. Like, we've been out the front of all these big, nasty problems. And guess what? We've learned from them, right? We've learned that just because you see a beautiful result, what is Russ's old adage? He says, if you see a beautiful result, treat it with the same cynicism you would if it was a bad result. And I think this is a lesson we've learned as a field. A lot of other fields haven't had to do that as publicly. But the good news is, if we take that with us and we start to develop new methods, they're going to be battle tested because they can't fall prey to all those old problems that we have had before. You've got to be better. So
Yeah.
it's a harder time, but I think it's a richer time because the methods that will come out will be trustworthy because they just simply otherwise won't pass muster.
Yeah.
So I think it's an exciting time to be a student if you've
Yeah.
got the gumption.
Yeah, and yeah, exactly. I completely agree, and I think it's important, right? I mean, it's hard for a student to do this because you have to learn a lot. You have to kind of get a sense of what's going on. You have to be confident enough to, to question the technique, and just because other people have used it, it will bring you down. Like you said, it might paint you into a corner, and not only the technique, but also, um, I remember Russ was talking about this a long time, a while ago, and he still is the idea of terminology. Like, obviously it's easy with the brain, is you tap your fingers, you see motor cortex, but then terms like attention or, uh, and I thought, okay, so now, I was reading your papers and I'm thinking, okay, so you have these cortical thalamic loops, and you say that the thalamus sort of exercises control over the brain states in some sense, and then, I'm thinking, okay, how does attention play in? Yeah,
So in all cases, when you throw something like that at the wall, you have to define what it is you're throwing first. And I think this is one of the challenges, right? Attention is this. What did William James say? We all know know what it is, but none of us can define it.
right.
We know it when we see it. Look, I try to stay away from terms like that, but I'm deeply interested in them. I really took Russ's lessons to heart when I was a postdoc with him. He was a great mentor for me. He hit me exactly the right time in my career where I could do a bunch of things in imaging, but I don't think I could do them at a level that was sophisticated enough that I could really trust exactly what I was seeing. And I went to him for methodological expertise, and that was extremely helpful. But the thing that really stood the test of time for me as well, which I didn't anticipate, was really a kind of philosophical appreciation for just how much the field has made a little bit of a kind of map territory mistake, I think, where they've said, I care about working memory. I've got a task that other people have used to describe working memory. Let's say something like the NBAC task. Therefore, anything that I see is working memory. And the cognitive neuroscientist puts their hands up and says, well, actually, the MBAC task has got active working memory where you have to expunge the previous trial from your memory, but it's also got to maintain that so that when it becomes the previous N trial, now you have to have it as an active component. You've also got to attend to the screen. You've also got to resist the distraction of the outside of the screen and ignore the noise.
Yeah.
You've also got to respond in a motoric way, right? It's got all these different components in it. And so I think sort of pulling back and saying the terms that we've used have not by anyone's kind of nefarious intention, have just become inextricably intertwined with the maps that we have. And I think if we can pull back, and then I think if we take Yuri Bojaki's impetus here and say, let's see what the brain does, and then later on, let's see whether or not that the set of brain constraints provide a better set of building blocks for us to understand the functionality of the system.
Yes.
And my guess, if I was going to fast forward and you wanted me to speculate, is that terms like attention, let's say overt attention, and perceptual consciousness have been conflated in the literature in lots of different cases. Things like covert attention and working memory are probably related together in other kinds of ways. And I don't think that these Venn diagrams will be perfectly overlapping. There's going to be really important contextual features that differentiate them. But my hedge is that the kinds of frameworks that we'll develop that build from the neurobiology and this weird and wonderful multi-scale system that forms our nervous system in our body, in our environment, will be such that we can then say later on, oh, what we really should have been saying was amplification of a signal was the really crucial thing. And if you do that in this context, we'd call it attention. If you do it in this context, we call it consciousness. And if we do it in this context, it's called working memory.
Yes.
And the process now becomes the kind of organizing principle rather than the outcome, which is, oh, hey son, why didn't you take out the garbage? Oh, I wasn't paying attention to you. These kinds of these psychological labels that we give to experiences. But that's absolutely looking at things through my kind of neurobiologically rose-colored glasses.
Well, I agree. I hope that the terms will sort of dissolve away and
Mm,
into a more sophisticated, nuanced sort of view of, dynamic systems sort of description in that sense. And that's the idea. And so, and I mean, the picture you paint, and I think that um, the type of research you're doing, I really do think that it's powerful in the sense that you are, forging new ground in methods, and then, bringing it back to systems neuroscience, and sort of right asking questions in just a different enough way that's sort of forcing fMRI out of its ruts of going down these paths. And yeah, I mean, sort of like, so I actually think like things like attention might, or consciousness or whatever will be similar to terms like, in physics, like ether,
yeah, yeah.
or things like that. So
My
um,
favorite's the difference between organic and inorganic chemistry, and there's still different lessons that are taught in universities. Almost every university splits into organic and inorganic chemistry, but that's based on the fact that they thought there was something in the chemistry at the chemistry level that differentiated life and non-life. And in the end, they were like, oh wait, they've all got the same atoms and covalent and ionic bonds and different things. It's just the way in which they're organized, I think, is probably our best hypothesis now that gives rise to the difference between life and non-life. But I think you're right. I think that a lot of these distinctions feel like they resonate with us as a really crucial way to carve up the world. But natural selection could care less about whether or not we can make sense of the world. It just finds good enough solutions to the problems at hand, and like water on a crack on a pavement finds all of the cracks. It'll
yeah,
just find the ways that it can solve problems, and some of those will be clearly dissociable. There'll be very different things, A versus B, and other times it'll be really weird spectra that go from A to B through all the different iterations between. And if you just stumble across something that's half A, half B, how do you categorize this? It's really difficult. Well, guess what? Natural selection doesn't care. It just did it
yeah.
because it worked. So I think we have to keep that in mind when we're trying to understand how this stuff fits together.
So that actually brings to mind another question. So, I mean, these questions are very different than sort of how I was planning this, but this is great.
Oh,
This is
sorry.
awesome. No, actually, it's better. um, uh, yeah. Uh, right. In terms of talking about natural selection, that's actually another thing that people are trying to find. I mean, it's sort of like, do you think that that there'll be, I think what we're trying to get at through modeling in some sense, and this is, you try to do this with physics and modeling to some degree, is try to go beyond observation and just sort of empirical observation to sort
Yeah.
of bring it together and sort of find principles, and the
Hmm.
more broad principle like natural selection that explains things, the better it is. And do you
Yeah.
think that uh, we'll ever get to that point where we can actually have like a set of principles that are driving the brain versus how the brain is organized versus just a bunch of hacks that are sort of used, and
Mm.
maybe there's a principle of how the hacks are used. I mean, it might all combine in some sense, but um,
Yeah. It's a great question. And it's one I spend a lot of time thinking about. I think I'm going to side with Dan Dennett here, who unfortunately passed away recently. He was a big inspiration for me. And he had this really lovely sort of line of reasoning in his book Consciousness Explained, where he said one of the challenges that he finds as a philosopher is that people want really magical explanations for things like consciousness or the immortal soul, these kinds of things. But at the end of the day, one of the lessons we can take from the natural world is that there's often really simple explanations for things that you just never really appreciated. And once you see them, you're like, oh, is that all it was? But right, and that's one way you could think about it. But another way to think about it is like, man, that's amazing. That something that simple can explain that
yes,
much. So natural selection is a really beautiful example.
yep,
Just recently, my kids were learning about Newton in class, and they were like, "Oh, so what? The apple fell on his head. Surely he'd seen apples fall before." And I said to them, "Yeah, but he actually figured out at that moment. Oh, wait a second, gravity is just falling." Right? It was that triggered for him that gave him the basic principle that explained all this
yes,
other beautiful mathematics. And
yep,
I think that we always have to be, in my opinion, this is where I get a bit romantic. I think searching for these kinds of compressed analogies and abstractions that help us understand the system. I think that the answer is yes, that we will absolutely develop these over time. And I think there's already heaps of them out there if you know where to look. Um, for people that are interested, Paul Chisek, University of Montreal, had a beautiful paper a number of years ago on phylogenetic refinement, where he essentially goes back over the cladistics tree, right? All of the different model organisms that we've been able to track over phylogeny. And he looks at how when they diverge from their common ancestors. And he looks at their brains and he essentially says, what was it about their brain at, say, point A that was different from point B? And what was it that the animals at point B could do that the animals at point A couldn't? Like, what was he almost tried to infer what was the pressure that caused that change and what was the brain structure that could change in order to fulfill that pressure. And Paul himself will admit that it's not a complete map because this is a really, really difficult thing to do. You're essentially going back in time and really desperately wishing that a different type of an animal left a better fossil record than the one that you have, because it's really difficult to make these kinds of inferences. But the cool thing that comes out is that you can say things like, oh, at one point the brain had no bilateral symmetry and now all of a sudden it does. And what the animals could do is now they can escape rather than just go towards things. Later on you can say, oh, now all of a sudden there's a colliculus. What can the animal do? Now they can detect things in the world and move towards them while also keeping track of the rest of their environment. And they can do these really complicated behaviors. And so you can start to kind of like build up this explanatory map where really simple, almost, you want to call them almost mechanical changes in the underlying neurobiology gave rise to changes in the animal's behavioral capacity. And it obviously gets really, really, really complicated the further you get towards us, because culture and language has just had such an acceleratory effect on everything that we're capable of doing. And I mean, if you look at our brains versus a common, close relative, a gorilla or a chimpanzee, they really don't look all that different, right? There's some parts that are a bit expanded. The lateral cerebellum's a bit bigger, the medial thalamus is a bit bigger, the pons a bit bigger, the cerebral cortex. But it's almost like the software that's running on that hardware is just completely changed. Now you've gone from running an old Atari game to running some of these modern, Unreal Engine type games. And the capacity for interaction between different people has gone up exponentially, maybe even super exponentially. So I think a lot of the answers we're going to look for, that are going to be the most satisfying, are not going to be just brain stories on their own. They're going to be, how does that particular set of brain constraints interact with this new virtual world that we're in, and how does that provide constraints or augmenting capacities or whatever on the ability to do that thing in that space. And so you're going to need help from sociologists and anthropologists and linguists and all these different specialties at the end of the day before you get those really rich understandings of why did I swear at my partner rather than compliment them in that moment that led to the fight. These aren't going to be like, oh, your cerebellum was wonky. It's going to be because you've got this particular software running on this really rich hardware system. That's going to be where the explanations will come from.
That's interesting. That's actually really right. I mean, that's actually right. I'll take a look at Paul Chisek's paper and
I can pass you a link. It's a beautiful paper.
Yeah, no, it definitely, and it reminds me. I mean, a very low level. I mean, still like sort of a mystery is, the whole idea of reading. Reading text. We never evolved to read text, and and it's something that we've, it seems that it comes sort of naturally, and we learn, and that's actually another aspect that I wanted to talk about. Sort of like automaticity. Maybe it is sort of a, what part of a brain that was evolved to do something is doing the reading is actually, and obviously language neuroscientists are exploring this well, but that's reminds me of that. So
so the automaticity part has been of my group. It's interesting. It doesn't necessarily flow on from the other work that we're doing as cleanly. I think if you start asking how do we track brain dynamics, well, nonlinear dynamics is really helpful, and how could the brain do it? Well, neurobiology makes sense, but the delegation thing or the automaticity question is something it's really bugged me for a long time. And I remember thinking about this actually back in medical school, that I would get so good at doing, taking blood from someone, and putting in a cannula, that if I thought about it, actually did worse. I was like, what is this? What is going on? Like, why is my focus hurting me here? I need to be talking to the person. I do it effortlessly. And I thought, that's so weird. Like, why would our brain do that? And then everyone has that experience of driving home, and then all of a sudden you go, well, how the hell did I get here? I don't remember any of the path, but I was having this lovely thought process in my head. And so I think this notion of how we do things without thinking about them is a really sort of massive kind of elephant in the room for us as neuroscientists that we almost never talk about. In cognitive neuroscience, almost all studies, and it's not exclusive, but it's the vast overwhelming majority, are focused on deliberate conscious processing, right? You are conscious of stimulus X and therefore you push Y. And there's definitely some stuff on implicit learning and things that gets away from it, but almost always we're focused on deliberate processing. And we haven't really asked, I think, the right kinds of questions for getting at not just how do we form habits or automatic processing, because those two things aren't necessarily exactly the same, they're often overlapping. But we haven't really thought, how do we even enact relatively automatic processing? How do we understand that? And so I've got a couple of papers. One I actually wrote with my father, who's an evolutionary biologist, because he asked me, how on earth is it that your son, my son Tyler, was about 12 months, 13 months at the time. How can he walk without stumbling? Because about a month ago, he was just like, he looked like a drunk sailor, coming off the ship. It really sent me down a massive rabbit hole. How on earth, what has changed? It's a month of time, but something in his brain's developed. And to cut a long story short, I think the culprit here is an area of the brain that we almost always ignore, which is the cerebellum. And it has over half the neurons in the adult brain. It does this incredibly interesting thing, which is it takes copies of all of our motor outputs. The main cell that leaves our cerebral cortex, in fact, it's the only one that leaves and goes into the spinal cord, is a layer 5B pyramidal neuron. Okay. And as on its way out to the spinal cord, it sends a little projection off to the pons, this big bulge you'll see if you look at a slice of the brain. It then synapses into the cerebellum, massive expansion, massive collapse, and then right back to the thalamus and to the cortex. And we're working on some ideas at the moment that essentially argue that algorithm is really the kind of algorithm that you'd want. You want that kind of architecture to essentially learn in any given moment when I make an action, what should come next and what should come next quickly, so that now I don't have to make any further guesses about it. That's just what I assume is happening. Yeah. If you do that well enough, now all of a sudden my deliberate conscious system, maybe that's riding in the corticothalamic system on top of that, can focus on something else in the environment and then allow that to run almost automatically underneath. So it's almost the perfect implementation of this algorithm.
That's interesting.
And so that's a huge, massive rabbit hole that we're in the moment. But it does, I think, then kind of turn back around and connect with the other parts of the lab, because this is a deep neurobiological study. It's about dynamical systems, interactions, but now almost dynamical systems running in parallel. So rather than thinking about the nervous system as this kind of N of one, what am I conscious of looking around the world like a little spotlight? Now you've got that spotlight, but it's running around with a whole bunch of processes underneath it that are anticipating this, that are holding this steady, that are making sure that you don't fall down, that are, keeping track of how long you've been talking or whether or not you're coherent or not. All these other features are kind of around that can then come up to conscious awareness if they fulfill some criteria. They get some little boost, but are otherwise running along under the surface. And to me, that's a much richer explanation of what it's like to be me in the world. I'm not always conscious of everything, but it all runs. Right. And I think we need better explanations like this in order to understand how our brain works.
Yeah, no, actually, there's so much there that you were mentioning in terms of what most of our processes are below our conscious awareness, and our conscious awareness only serves a purpose. It serves a survival purpose, an evolutionary purpose that
it's useful for something.
Things changing, but it's interesting when you mention the cerebellum, because it brings to mind. Talking to a colleague of mine, he was talking about, once again, back to modeling. People are attempting to model, the cortex as a deep neural network, and it seems that doesn't really apply. It's more of a sparse, shallow network. But the thing that can be, it seems like, modeled as a deep neural network with multi-layers is the cerebellum potentially. And that's how it does its processing, in some sense.
It's a great insight, and it's a one that we're thinking a lot about at the moment. We've done a little bit of neural network modeling, but usually to kind of constrain our sort of hypothesis space about how a task could be solved. So, you train the RNN to do a task, and then you interrogate it. And the beauty of that is you can take out any cell you want, or you can turn down the volume or the gain or whatever. And you're like, oh, it actually does the thing I expect. Is that in the data that I actually have as well? And that's a really fun experience. using an RNN or using RNN or a transformer or a column gen or something like that as a stand-in for the kind of neurobiological feature of interest is a bit of a trickier space. And the reason I say that is that the goal of modeling for me is not necessarily to make the best prediction of the data going forward. It's to understand why I was able to make the best prediction or not.
Yeah. Yeah.
And part of the challenge we have, I think, at the moment is that there's been this real divergence of fields where the machine learning field has gone, we can do really cool stuff.
Yep.
Just let us go. And it's just accelerated on itself. It's almost more like an engineering challenge, right? Like, can I learn all of the different ways that alpha proteins could be folded if I knew their primary structure? Could I anticipate what someone would write? To me, there's a kind of a thin line there where on the other side, the scientist says, why was it that, or how was it that this worked? And they go, but if we put more layers in, it does it better, you know, or if we use this gradient descent, it does it better. And I say, okay, that's cool. It's very useful for what you're trying to do. But for me, I want to say, what was it about the molecular layer interneurons or, the Purkinje neurons or the cerebellar nuclei that led to that function or not? Because then I want to be able to go back to my collaborators who work on these problems and say, can you please, pretty please, optogenetically silence this nucleus while the animal does this task? Because we think that the animal won't be able to grab the pellet anymore. And if I go down the deep neural net route, sure, I've got a fancy, shiny, thing that looks like a cerebellum, but I don't know why. And to me, that's not why I'm in this game. So I've avoided that for now. But no, I don't begrudge people to go down that route. It's a fascinating route. It's just not the one that we work on.
I mean, that right. I mean, hopefully that the two fields will sort of talk to each other more. I think they can inform each other to some degree. And like you said, keeping your eye on, understanding the principles behind it, understanding the why, understanding the neurobiology and whatever. And also, maybe designing better intelligent systems if there's some sort of elegant algorithm that the brain is using that is outside of what we're thinking of as well. So hopefully, there's emerging.
I think there's ways. There's ways that we can merge together. I think, again, there's a kind of underlying theme to our conversation today, Peter, which is that the dialogue and the communication is incredibly important. And I think one of the things I tried to underline in my talk at HBM was the kind of deep reverence that I have for the specialties that we have in our field. And I remember just being absolutely overwhelmed and terrified and imposter syndrome through the roof at my first 4 or 5 HBMs, because you're wandering around these poster halls and people are just like spinning the most incredibly complicated analyses and thoughtful experiments. And you're just like, how on earth could we ever connect with this stuff? But I think it took me working with Russ to realize that nobody does any of this on their own. What happens is you collaborate with really, really brilliant people. You find that workspace together, and then together you do the work at a high level that would pass the muster of all the other different individuals. And so it's not about an individual, it's about a collective. And I think we've got a really great opportunity at HBM to essentially kind of leverage that great kind of collegiate atmosphere we have. We're very sharing, where we've got everyone will put their code on GitHub. They're quite happy to explain to you why they chose parameter X rather than Y. And I think I would really encourage the students out there, if you're interested, don't hesitate. Reach out to people. What's the worst they can do? They're going to ignore your email. Okay, cool. There'll be another person who's worked on that algorithm. Reach out to them. It's a really, really constructive community that I think believes in the strength of the community over the strength of the individual. And I really wanted to just underline that. Any student that's out there feeling imposter syndrome, we all feel it. It's absolutely part of it. And it's actually a sign that you're around a bunch of intelligent people and you're going to do cool stuff. So it's a good thing.
And the fact is, none of us really know completely what we're doing. I mean, we're sort of doing the best we can, and
exactly,
searching at the edge of what we know, and that's what we do. And
exactly,
So this is approaching. You've gone over an hour, but I have to sort of ask one other aspect of this. And so, what do you think? One of the aspects of what I'm working on and what the field is sort of trying to push a little bit is the idea of going to very high resolution fMRI and getting layer information. And as I was reading your papers, I'm like, oh wow, I'd love to try to do something like this, where you look at the projections and you try to see if these changes in states, if there's middle layer activity versus upper and lower layers to some degree. Is your sense that would be informative or?
Absolutely. So in the middle of the pandemic, we kind of internally felt even further away from the rest of the world. Sometimes that's a good thing, to be far away from the rest of the world when things are going particularly crazy. But during the pandemic, we felt the kind of stings of it a little bit. And so one of the things we did is we reached out to a lot of people that we really admire and had them come and give us talks about their work. And so Renzo Huber, I know you've worked with quite a bit,
Yes.
gave us actually 2 talks. We invited him back because the first one was so exciting and mind-blowing. And we also had Emily talk about some of her work on the layer stuff with the working memory task. So deeply interested. According to Renzo, when we chatted a couple of years ago, it's a bit of a technical hurdle at the moment to get really good signal to noise in the cortex, but also be able to track simultaneous activity in the thalamus or ball in the thalamus. But he said, watch this space and we'd love to chat. I think you're totally right that getting the resolution of supragranular, granular, infragranular is deeply informative. The only part of it that I'm nervous about is that one of our kind of main collaborators in that kind of physiology world, Matthew Larkham, who works in Berlin at the Humboldt University, he studies this, in particular, this really fascinating cell that I mentioned before, the main output of the cerebral cortex, a layer 5B pyramidal neuron. And it's called a layer 5B pyramidal neuron because it's a pyramidal neuron that has a cell body that sits in layer 5B, which is an infragranular layer of the cortex. If you look at the topology of this cell, though, actually what happens is it actually has this really, really long apical stalk that goes all the way across the granular and infragranular layers to the layer 1, the crowning mystery of the cerebral cortex, and then fans out like a massive tree in a forest, a massive canopy. And it receives all these huge inputs to that area. So now we've got a cell whose function depends deeply on both infragranular and supragranular activity patterns of inputs and outputs. And it turns out that context-dependent inputs, so if you have input that comes into the cell in the base layer, it actually sends a traveling wave of calcium up the stalk so that any input that comes in next, it's called a back-propagating action potential. Any input that comes in next now goes back down the stalk and causes the cell to burst fire. It just goes crazy. It just starts like alarm bells ringing, just pumping action potentials. And Matthew has some really lovely work showing that when they give mice different anesthetic agents, this back-propagating action potential is blocked. That's a beautiful Cell paper. Suzuki's the first author. And then in another line of experiments, they've actually shown that if they train a mouse to actually detect a whisker twig and then measure these cells in their barrel cortex on the opposite side, they have them lick when they're trained to lick when they notice the twig. What they can actually do is they can tickle their whisker lightly, and they can then increase the intensity, and they can actually map out the psychometric curve of the animal, and they can actually show that these cells in the barrel cortex show that burst firing in the same proportion. They call it a neurometric function. So now what Matthew's been able to do is link this particular cell type, which is deeply interconnected with the thalamus and the rest of the brain, to both intransitive changes in consciousness, change in conscious state, but also transitive features of consciousness of the whisker twig or not. So this to us is a really, really great example of a cell that we deeply want to understand better, but has this weird anatomy that's sitting across different layers. And it's hard to know whether or not blood flow to those layers would be impacted by spiking. And so it's absolutely, my answer is absolutely yes. But my answer is we need to talk to people that do this stuff and think carefully about how to do it properly.
And like everything, we have to be very careful about how we interpret it. And you might see layer modulation, but it might be very different than what's actually going on. I totally agree with that. But we'll be trying first getting the technical aspect, and then trying.
We should chat.
Yeah, definitely.
Awesome.
So, all right. Well, I think that's a good place to end right here. But I really appreciate you coming on, and keep on doing what you're doing, and hopefully we'll all look at your work as an example and be inspired to do similar yet different sort of work. And thank you.
Thanks, Peter. Thanks for having me.
Neurosalience is brought to you by the Organization for Human Brain Mapping. This episode was produced by Alfie Wearn and Karthik Sama.